Viruses. What are examples of DNA and RNA viruses, and how do they differ? Are you sometimes confused and overwhelmed by the large number of virus types? Let's try and get around this by grouping viruses according to their genome and understand their corresponding characteristics. During transmission, the virus particle contains either DNA or RNA as genetic material and is surrounded by a protein shell, called a capsid, and sometimes an envelope. Once the virion penetrates the host cell, the viral genome is unpacked, resulting in the hijacking of the host cell's transcription and translation machinery. The interaction between the host and virus depends on the nature of the viral genome. Therefore, it helps to distinguish viruses according to their genome. The virus classification system based on genome structure is the Baltimore classification. It was developed in the early 1970s by the Nobel laureate David Baltimore. According to this classification system, viruses are separated into 7 groups. Let's start with a relatively simple case. A viral genome similar to the genetic material of the host, double-stranded DNA, or in short, dsDNA. Many viruses belong to this first group, including adenoviruses, herpes viruses, pox viruses, polyomaviruses, and papillomaviruses. Most viruses in this group are usually required to deliver their genome into the host nucleus, where it can be transcribed by the host's infrastructure. It should be noted that the viral DNA stays as a separate module and doesn't fuse with the host cell DNA. Let's use another simple example for RNA viruses, namely a virus with a positive sense, in short, plus, single-stranded, in short, SS genome. The viral genome can remain in the host cell's cytoplasm and either functions as messenger RNA and is translated into protein or is transformed into messenger RNA. RNA viruses require their own RNA polymerase to replicate their genomes, as the host cell is unable to replicate the RNA template and doesn't possess a suitable enzyme. Positive-sense, single-stranded RNA viruses belong to Group 4 of the Baltimore classification system. Notable examples include the rhinoviruses, the primary cause of the common cold, as well as the hepatitis A and C viruses. Viral genomes are diverse in size and structure and can consist of positive-sense, single-stranded DNA, as found in parvoviruses, classified as Group 2 viruses. As host cells can't use single-stranded DNA for replication or transcription, these single-stranded viruses also transport proteins required for converting their DNA into double-stranded DNA. This is also the case for the hepatitis B virus genome, which is circular and contains gaps. These gaps are due to the incomplete synthesis of the positive strand. The positive strand gaps need to be filled in accordance to the intact negative strand before DNA processing in the host cell nucleus. Hepatitis B virus belongs to Group 7 of the Baltimore classification system, which was only added to the system at a later stage as the genomic nature of the virus was unknown at the time. Viral RNA is comparably diverse and can also be double-stranded. A notable example are the rotaviruses, which fall under Group 3 of the Baltimore classification system. Yet viral RNA can also be single-stranded with a negative sense, as found in influenza, Ebola, and rabies viruses. Negative-sense viral RNA must first be transformed into positive-sense, single-stranded RNA before translation into proteins by the host. Interestingly, some positive-sense, single-stranded RNA viruses, such as poliovirus and Zika virus, go through a stage in which their genome is transformed into negative-sense RNA before being transformed back to messenger RNA. A very special ability can be observed in the retroviruses, which include human immunodeficiency virus, in short HIV. The name retrovirus indicates that its positive-sense, single-stranded RNA genome is converted into DNA. This process is mediated by an enzyme present in the virus termed reverse transcriptase. The viral DNA is permanently integrated into the DNA of the host cell, which can be transcribed upon gene activation. Retroviruses belong to Group 6 of the Baltimore classification system. So what are some advantages of having a DNA or RNA genome? DNA genomes are generally larger than RNA genomes, meaning that the information storage capacity is greater. This is beneficial as viruses with a large genome are usually less dependent on their host. On the other hand, DNA replication is much slower than RNA replication. But most importantly, DNA viruses are confronted with many elaborate repair mechanisms in the host cell because host DNA is intended for the long-term storage of genetic information. In contrast, the host cell's RNA is a short-lived molecule and consequently more likely to be degraded rather than repaired upon damage. Therefore, there are no elaborate repair mechanisms in cells enabling RNA viruses to mutate at a higher rate than DNA viruses. The transcription error rate in DNA is approximately 1 error in each 10 to the 7 nucleotides, whereas in RNA the rate is 1 error in each 10 to the 3 to 10 to the 4 nucleotides. High mutation rates are clearly advantageous in viruses, as can be seen for the influenza viruses, which mutate so quickly that the vaccine offers limited or no protection in the given season. This is also reflected in the eradication of smallpox. Eradication was only possible by extensive vaccination of potential human hosts, combined with the virus's inability to evade the sudden emerging immunity through mutation. In the next episode, we'll be taking a detailed look at the different viral life cycles of DNA and RNA viruses.